Hello, CHY 107. Here we are once again for a discussion of a certain type of biomolecule. This lecture, we're going to start to focus in on proteins. Now, proteins are another term that really kind of there's quite a difference between its everyday usage and what we mean when we talk about proteins in chemistry. When you think everyday usage, you think dietary proteins. I get my protein from meat, or from a vegetarian, I need to get my proteins from other sources, some beans and not beans, but sorry, some just different sources of my dietary protein. And certainly dietary protein and the proteins that we do get from our diet are a big part of proteins in general. But proteins are, again, just another class of molecule. And it turns out that from a chemical perspective, and especially from a biochemical perspective, that proteins are some of the big workhorses in our body, taking on tons and tons and tons of different roles. And there are lots, I'm talking thousands, tens of thousands of different types of proteins in our bodies, all performing different roles. And so that's what we're going to focus in on today is the basics of the class of the compound, or the class of molecule called proteins. In order to discuss proteins, we need to discuss the basic building block of proteins, which are another term that you're probably familiar with, amino acids. So this is another term that you've probably heard before. Now, amino acids, so have the basic structure here of a carbon in the middle with one hydrogen coming off, but then the three other groups, we always find an amino group, hence amino. We also always find this carboxylic acid group, hence the acid. And so that part of an amino acid never changes. Amino acids always have this basic structure here of a carbon, a central carbon, with amino group, a carboxylic acid group. But then this side chain here can be different, and we're going to take a look at that in a little bit. So different amino acids can have different side chains coming off here. At a physiological pH, about 7.3, the amine and the carboxylic acid group are both ionized. And so, in other words, and we'll see some pictures of this in a minute, but just to bring us back to our basic structure. At a physiological pH, instead of NH2 for our amine, it's actually an NH3, which would bring this N to a positive charge. As well as on the carboxylic acid side, this hydrogen is actually removed. So this is an O minus instead. And so at physiological pHs, we have both a positively charged amino side and a negatively charged carboxylic acid in the amino acid. So since there's both a positive and a negative charge present in the same molecule, that does mean that the overall net charge is neutral, but we call that a Zwitter ion. And so we can see this here, our Zwitter ion form. At lower pHs, we actually end up with the acid having its hydrogen on there, and the overall form would be a cation with a positive charge. At higher pHs, we still have our negatively charged oxygen on our acid, but the amino group is no longer ionized. And so that would give us a negatively charged anion. And that we're going to see where that comes into play in a little later on. But at normal physiological pH, the form of amino acids is this Zwitter ion form. So amino acids, as I mentioned, have differences in that R group, which is bonded onto the carbon. That's the only part of amino acids that will vary. So it's the distinguishing feature of different types of amino acids. All amino acids do have a name and a 3-letter abbreviation which distinguishes them from others. And here are just 3 examples here. So again, we can see in all 3 of these, we have valine, tryptophan, and cysteine. We can see that this basic structure here, our carbon group with our carboxylate group and our amino group, that doesn't change. That is always the same and consistent through all of these different amino acids. What changes is this side chain. So valine has this branching carbon chain here. Tryptophan has this carbon group with then these two different rings with a nitrogen thrown in there, whereas cysteine has a carbon group with a sulfide group there. And we can see the three-letter abbreviations, VAL for valine, TRP for tryptophan, CYS for cysteine. Incidentally, yes, this is the tryptophan that some of you have probably heard makes you sleepy after Thanksgiving dinner. I hate to break this to you, but that is a myth. Yes, tryptophan is something that can make you sleepy. I forget what the actual numbers here were. I looked into it at one point, but it would take something like eating a pound of pure tryptophan to actually have a sleepiness effect on you. And so the tryptophan in turkey is not what actually makes you sleepy. It's the fact that you just ate 3 pounds of potatoes, turkey, and gravy, and you followed it up with a whole pie. Then you fall asleep watching the Packers. Or, sorry, not the Packers. Sometimes it's the Packers, but you fall asleep watching either the Cowboys or the Lions. I don't know how I just made that mistake. But anyway, it's not the tryptophan. It's the fact that you eat so much damn food. And then your body is busy trying to process that. Anyway, and so amino acids differ by that side chain. And so here's several examples. And so those side chains can be either polar or non-polar. So here are all the different non-polar amino acids, glycine, alanine, valine, leucine, and so on. And you can see them all here. We also have, and this, by the way, it might be tough to see on your screen or in the presentation. This is on page 604 in your textbook, is a nice chart of all the amino acids. And I'm just pulling these images right from there. And these are all the amino acids with polar side chains. Some of them do fall into, touchy mouse today, some of them do fall into either acidic or basic categories if they're polar, and some of them are neutral. Things like aspartic acid, glutamic acid, lysine, arginine, histidine. Here's our cysteine that we talked about, serine, asparagine. So all sorts of different types of amino acids. You are going to need to memorize the side chains on all 20 of these. Did that cause any heart attacks? Just kidding. You don't need to memorize them. On quiz or in your final exam, I will allow you to use this same image, basically. I will have this set up for you and ready to go in your allowable materials. So you definitely don't have to memorize them. You just have to look on the chart to try to find them to see what those side chains are so you can identify them. All proteins in nature are constructed from these 20 amino acids. And so and that's another just factoid to know, is that there are 20 naturally occurring amino acids. If we counted all the amino acids on these two different charts here, there are 20 total, and that's all. There are only 20 different amino acids that are naturally occurring. 11 of those, our body can make them. The other 9 must be supplied through the diet. And just like we talked about when we were talking about lipids, the meaning of essential fatty acids means that those we have to eat because our body can't make them. We have to get them through the diet. Same with these amino acids. The 9 amino acids that we must get through our diet somehow, those are known as the essential amino acids. And so, and our body will need them for various things, which we'll see in a few minutes. So our body might need some of these, those 9 essential amino acids. And so we have to get them in through our diet. Animal sources, and this is one of the reasons why it's so important to have protein as part of your diet. Because as we're going to see, what we're building up to is that protein and proteins in general are made up of amino acids. And so one of the reasons why it's so important for us to eat protein is because that will give us these 9 essential amino acids that we need. And so our body can then break down the protein into its amino acids and use these for various things that it might have to do. Animal sources will provide all of these for us. plant sources will generally need to be combined in order for a person to get all the different amino acids in one meal. And so this is one of the reasons why it is more important for vegetarians to just be a little more mindful of what they are eating and make sure that they're getting all of the essential amino acids into their diet, because certain plant protein sources will have some of the essentials and others will have others. And so they've got to mix that up a little bit. But people who do eat animal protein as well, the animal proteins will get them all the essential amino acids that they need. So what we can do is we can start to take amino acids and bind them together. And so it's called a dipeptide when two amino acids, when two of them combine. And they're going to do so by having the amino end of one combined with the carboxylic end of another. There's our amidation reaction. So remember, we looked at those a few weeks ago. This is one of the ways that one of the things that we see that's so important is that we end up getting a new amide bond. So we form an amide, and so we can see our amide right here. Here's our C double bond O over to a nitrogen. So there's our amide bond. And so that happens. So the two of them join up through this amidation reaction to form a dipeptide. And we can see this dipeptide right here of two generic amino acids, amino acid 1 with whatever its R group is, amino acid 2 with whatever its R group is. Now they are joined together by way of a peptide bond. Polypeptides contain many amino acids. Proteins are very large polypeptides, which serve some sort of biological function. By definition, the end of the peptide that has the free amine is known as the N-terminus. And so here's a polypeptide made up of 3 different amino acids. We have alanine, glycine, and valine here. And so here's our polypeptide, and you can see a ball and stick model of it here. So here's our polypeptide, and we can see that alanine has the free floating nitrogen, the free floating amino group with nothing attached over here. So we call that the N-terminus. Then over here, the valine, the end of the valine ends with our carboxylic acid group, and we call that the C-terminus. When we're listing the amino acids that are in a polypeptide, we always list them in order from the N-terminus to the C-terminus. So no matter how you actually see it written out, if you saw this flopped around, you would still write it alanine, glycine, valine, because you would see that the alanine has that N-terminus on it. And so proteins are composed of from anywhere from about 50 to sometimes 27,000 even amino acids. Most proteins contain less than 2,000, so somewhere between 100 or so up to 2,000 or so amino acids long. Some proteins contain multiple polypeptide chains, structured and bound together in some way, and we're going to look at that structure in this lecture. Scientists do estimate there are actually, and what I said tens or thousands or tens of thousands, I was very incorrect. Estimates are that there are about a million different proteins in our body, all built from those same 20 amino acids and all coded for in our DNA. And that's a big part of what we're going to talk about in the next lecture when we talk about nucleic acids, is we're going to talk about the fact that that's one of the main roles of DNA, is it contains the code to build these proteins. There are 3 general types of proteins: fibrous proteins, globular proteins, and membrane proteins. Fibrous proteins tend to be strong and insoluble, and they usually serve a structural role. Things like keratins, which are found in hair or fingernails, feathers, horns, structures like that. We also have elastin proteins, provide flexibility to skin, blood vessels, heart, lungs. Then we also have collagens, which provide a little bit of rigidity to our connective tissues. Globular proteins are soluble polypeptides. They're able to dissolve in water, which get folded into spherical shapes. And we'll see some examples of these. Membrane proteins, we saw a bit in our last lecture on lipids. Membrane proteins are partly or entirely embedded into cell membranes, and they aid for the transport of things across the cell membrane. A transmembrane protein is one that crosses the membrane several times. So it might be kind of woven through the membrane a bit. And so let's talk about protein structure. There are 4 levels of structure to a protein. What we call the primary structure is the amino acid sequence. And so we see something like this here. So this would be a protein that it's actually composed of 2 different amino acid chains bound together. But we can see here, for instance, in this chain, it's composed of a glycine, what's ILE? Isoleucine, valine, glutamate. I don't remember what GIN is off the top of my head. But then a cysteine, alanine, serine, valine. And so we can see that we've got this just amino acid sequence. And then down here, phenylalanine, valine, asparagine, glutamine, histidine, leucine, cysteine, glycine, serine, and so forth. And so that sequence of amino acids, which amino acids are there and what order are they found in, is what we call the primary structure of an amino acid. The secondary structure. So what can happen then? So we've got our primary structure. And what can occur at that point? So we end up getting these long chains of amino acids. Due to the different R groups on those amino acids, those different R groups can begin to interact with each other in the same chain. That interaction with each other will lead to more shapes. And so you can picture maybe we've got this long chain that starts to curve around a little bit, and then some of the R groups begin to interact with each other and maybe form a hydrogen bond there. Or maybe form what we call a disulfide bridge, and we'll talk about some of these. And so the secondary structure is the folding patterns that can start to happen in some of these localized regions of this amino acid chain. And it's due partly from those interactions along the chain, but also partly from the interactions of those side chains with the surrounding environment, namely water. The 2 most common types of secondary structure are what we call alpha helices and beta sheets. An alpha helix is formed with hydrogen bonding between the carbonyl oxygen and an amide hydrogen, which is about 3.5 amino acids away. In a ribbon diagram, the alpha helix appears as a helical colored flat ribbon. Let's just take a look. And so here we see what we mean by an alpha helix. And so we can see our amino acid groups here. And we end up with this hydrogen bonding that's occurring between carboxylic acid groups. So we can see a clear one right here. So here's a carboxylic acid group of 1 amino acid, hydrogen bonding to the amino group of another amino acid within the same chain. And so we get this hydrogen bond that shows up, which ends up giving the whole chain, or this part of the chain anyway, it gives this part of the chain this helical shape, which we can demonstrate here. This is what we call a ribbon diagram. This particular protein is myoglobin. And we can see that ribbon showing up right here. And it arises from this hydrogen bonding occurring between the carboxylic acid group in one part of the chain and the amine group in another part of the chain. And so that gives rise to this helix. A beta sheet forms when 2 or more parallel, or what we call anti-parallel sections, fold in a pattern that looks kind of like a pleated skirt. This happens when we get hydrogen, again, same idea, hydrogen bonding occurs between an amino group and a carboxylic acid group in an adjacent chain. But it happens when we get more flat sections in the whole entire chain. And that we depict like this. And so we can see these beta sheets unfolding here, sort of one on top of another. And again, arising from some hydrogen bonding interactions. And so those are the two main types of, of secondary structure in proteins, either an alpha helix or a beta sheet. And so we start, and so kind of picturing where we're at, we have this long polypeptide chain, this long amino acid chain. The backbone in that amino acid chain is going to start to interact with itself at various parts, which then gives rise to some helix shapes or some of these beta sheet shapes within the protein. From there, we then have, sorry, and I was mistaken earlier when I said that part of that could be a result of the surroundings. The surroundings start to give rise to what we call a tertiary structure. And so we've got this long amino acid chain, parts of it coil like a helix, parts of it fold up like pleated sheets. From there, we start to get even more sort of organizing and rearranging of the whole entire structure into something for a shape. And what affects that is mostly electrostatic interactions between the R groups and the water surrounding the outside. And so if we have a very polar R group, that's going to tend to come to the outside and interact with the water. Non-polar R groups will tend to go around to the inside and try to get away from the water. And so we end up with this different shaping that goes on as a result of that. In some proteins, this also includes some things like a heme group, for instance, in hemoglobin. Hemoglobin is just a large protein which is responsible for carrying the oxygen around in our red blood cells. And so this heme group is also part of the protein, and that's going to affect the shape in there. And so as I mentioned, the fundamental driving force behind tertiary structure now is to try to minimize the interactions of water with non-polar R groups and maximize the interactions between the water on the outside of the protein and polar R groups. We also have additional interactions which will contribute, such as what we call disulfide bridges. A disulfide bridge is a covalent bond that will happen between two cysteines. So if you have two cysteines that end up in proximity to each other, remember cysteines have an SH, they have a sulfide group in their R chain. And so that sulfide group will end up bonding between the sulfurs. So that's hence the name disulfide bridge. We can also get salt bridges, which are essentially ionic bonds that happen. So a positively charged R group and a negatively charged R group, those will tend to stick together. And then the hydrogen bonding and dispersion forces between polar and non-polar parts of the chain. So already actually talked about these. And so here's where we can start to see some of that in action. Sorry about that. And so here's our general protein chain here. And we would see some alpha helices and some beta sheets within this chain as well. What we start to see is, for instance, this, and I don't remember what this one is here. I don't remember all the amino acids off the top of my head, unfortunately. But we see this one here, and we've got this positively charged NH3 group on the end, and we have our negatively charged carboxylate group at the end of this one. And so we're going to create a salt bridge there, an ionic interaction between a negatively charged species and a positively charged species. And so that's going to be an attraction. But here we've got dispersion forces that are trying to bring these together. Non-polar group and a non-polar group will have dispersion forces that attract them together. Here's a cysteine in this part of the molecule I recognize from the sulfur. Here's in the polypeptide. Here's a cysteine on this other part, and so those are going to form a disulfide bridge that will bring them together. Here's a hydrogen bond bringing them together. And so all these different interactions amongst the R groups will then give rise to different folding within the protein and another layer of structure. And so we'll talk about the fourth layer in just a minute, but let's just recap that. And so we have our primary structure, which is our amino acid sequence. We have secondary structure, which can arise from the backbone of our amino acid, giving rise to things like helices or beta sheets. We then have tertiary structure, which arises from interactions between our R groups, and that leads to what we call protein folding, and a lot of just sort of convoluted folding around and shaping of the molecule in general. Then we can have what we call quaternary structure, the fourth layer of structure. That arises from having two or more polypeptide chains interacting with each other. And hemoglobin is a fantastic example of that. And so here we have one polypeptide chain of hemoglobin, but the actual hemoglobin protein is going to be made up of 4 of those different peptides coming together and forming this large, big globular structure. And that's the final order and final layer of structure in a hemoglobin protein. And so we have these 4 different layers of various structure to proteins in general. Now, with all these different layers, it can be easy for something to mess with that and to start to break the structure down. And in fact, we do see that quite often. That's called denaturation. Denaturation of a protein occurs when there's some sort of disruption of the secondary, tertiary, or quaternary structure. When that structure breaks down, proteins can no longer perform their function. One of the main properties of proteins, one of the main things that allows proteins to do what they do is the shape. There's an expression quite often in biochemistry that's along the lines of form equals function, which means that the shape of proteins plays a huge role in their function. And so if something happens to alter that structure, the protein can't do its job. Denaturation does not alter the primary structure protein, doesn't mess with our amino acid sequence. And sometimes it can be reversible. Proteins can be denatured by a number of things. Heat up a protein, that can cause it to break down. pH changes, mechanical agitation, literally shaking it up, detergents, certain metals. These are all things that can break down either the secondary, tertiary, or quaternary structures. A denatured protein will lose its shape, and as I mentioned, will thus lose its biological activity. This is one of the reasons that it's so important to maintain physiological pH within our body. If pH changes, as what happens with either acidosis or alkalosis, which we talked about quite a while ago, what happens then is that the positives and negatives along some of our chains changes. And so where we might have a salt bridge that's occurring right here, leading to some of our tertiary structure, if we have acidosis, and so we end up at a higher pH, now we've lost our negative charge on the carboxylate group, so we can't have a salt bridge there. On the other hand, if we raise the pH, alkalosis occurs. Now we're going to lose our positively charged amino group. No salt bridge. And so that can disrupt salt bridges. We can disrupt hydrogen bonding in the same manner, sort of losing or gaining hydrogens from various parts due to pH changes. Denaturation actually leads to, coincidentally enough, my breakfast this morning. I had a couple of fried eggs for breakfast today. And so we see the native conformation. And so a normal shape of the protein in eggs, some alpha helices in there, some beta sheets. We heat it up. and that denatures the protein, leads to delicious, not sunny side up, delicious over easy eggs, slightly runny. Enzymes, something else that most people probably heard the term. Enzymes are nature's catalysts. They are proteins which speed up chemical reactions in our bodies. Every enzyme catalyzes a specific reaction, acting specifically on one substrate, on one type of compound. An enzyme, and one of the key factors of an enzyme is that itself is unaltered during the reaction, and we actually don't show it as a reactant. If we were to look at a reaction equation, we don't show the enzyme in there. If you see something that ends with the term ACE, that's an enzyme. And it's usually named after the substrate as well. For instance, the enzyme Sucrase. Remember, we looked at sucrose a little bit, looking at our carbohydrates. Sucrose is made up of a glucose molecule and a glucose monosaccharide and a glucose monosaccharide. And so the two of those together, that makes up sucrose. Sucrase is an enzyme which will help break those apart. Reaction rates for enzyme catalyzed reactions vary. So an enzyme can catalyze a reaction to happen once every 2 seconds to maybe even as fast as 10 million substrate molecules being undergoing the reaction every second. Let's actually look at an example here. And so this actually is a great example of how the sucrase enzyme that I just talked about works. Here's the enzyme. Here would be the sucrose molecule. And we have what we call the active site on the enzyme. Enzymes are often are referred to as having a lock and key mechanism because there's only one type of substrate that's going to fit into this enzyme. This active site will only fit this one particular substrate. So in the case of sucrase, the active site on that enzyme will only fit a sucrose molecule. And then it fits in there, and then the enzyme acts in some way to, in this case, break the two parts of it apart. And so this would be the two different monosaccharides that make up sucrose would be able to be broken apart. So as I just mentioned, so the substrate binds to the enzyme in a pocket or a groove known as the active site. Some sort of conformational, some sort of shape change occurs in the enzyme, allowing whatever it is that the enzyme is going to do to actually happen. And this selectivity is very important because there's going to be a number of different molecules in the area. But we only want the enzyme to act with that one particular molecule. And actually, we're not going to worry too much about the activation energy. Let's just go on and talk a little bit about enzyme inhibition. And so enzyme inhibitors are things that actually will bind to an enzyme to prevent it from functioning. So remember I talked about that active site being particular to one type of substrate. An enzyme inhibitor is something that might mimic that substrate and bind instead to the enzyme, which stops it from working. Two types of inhibitors. A competitive inhibitor simply binds at the active site, blocking the substrate from coming in. Non-competitive inhibitors bind somewhere else on the active site, but by doing so, they still prevent binding of the substrate to the enzyme. So competitive versus non-competitive, competitive binds right at the active site, non-competitive binds somewhere else, but the bottom line is they both prevent the enzyme from coming together with the substrate. And we can see that here. So here's what would be a normal substrate enzyme complex. Competitive inhibitor will come in and bind directly so that our substrate can't come in. A non-competitive inhibitor comes in, and we can see in this case changes the shape of the enzyme so the substrate can't come in. Great example of this, not technically an enzyme, but the same sort of competitive mechanism, caffeine and adenosine. Adenosine is a substance in our body that helps make us sleepy. And there are receptors in our brains to capture adenosine. And so when these receptors are getting filled with adenosine, we're sleepy. Caffeine competitively binds to those receptors, kicks adenosine out, and binds to that receptor so that adenosine can't get back in. Wakes us up, prevents us from being sleepy. So same exact idea. Like I said, it's not an enzyme. And so don't go take away from this thinking that caffeine is an enzyme, because it is not. But it's that same sort of competitive inhibition. So I like to use it for an example here. And this last slide is just our amino acids again. I just had them all in one place so you could see them all. And so, which reminds me, okay, I just want to make myself a little note of something that I am thinking. Okay, and so that is it for our discussion of proteins. When we come back for our next lecture, we will be discussing, we're going to go into some discussion on nucleic acids, DNA and RNA, and the role that proteins are playing in there. And so that is all for today. I have to go because it is definitely time to wash my hair. Getting pretty greasy, but yeah. All right. You stay classy, USM. Till next time.